phylogeny paper. At the beginning of this file, we have a commentary written by Trevor Price. I want you to read this first because it gives you maybe an easier introduction to the paper than jumping straight into the paper. It'll give you a little bit of background about the Hawaiian honeycreepers that didn't make it into the final paper, and a little bit of more information about how do you make this kind of a phylogenetic tree. So start here with adaptive radiations or something about finches, and read through. You'll note in this figure that there are a series of species that are not present in the tree that is in the next paper. So these species in red are species that are extinct now, and the ones in green are the ones that are still alive today. And the paper that you're going to be reading is one that focuses just on the modern species. The extinct ones are the topic of another paper that's underway now. So read through Trevor's introduction. Trevor's going to talk a little bit about the difference between gene trees and species trees. So if you look at this figure 2, you'll recognize a gene tree inside of a species tree, and you'll recognize when the gene tree and the species tree are different. This is something we talked about. So here we are on the paper, and I'm going to point out, after you read through Trevor's going to give you basically what I would do for a reading guide. So I'm just going to point out a couple of things that are specific to some of the figures and some of the details in the text that he didn't pick up on. So going through here, this figure, we talked briefly in class about the idea of a cloudogram, and what this is the type of analysis I did. It doesn't return a single best tree. It returns many trees that are almost equally likely. So in this case, we have all of the trees that came back as being highly likely. These are 19,601 trees, all shown over the top of each other on the left. Why am I doing that? I'm doing that because I want you to see that all of those trees vary slightly, but mostly they vary in branch length, so in timing of the divergences. So where the lines are fuzzy, that means that we don't know, for instance, if this right here is where this divergence happened or if it's slightly further back in time, right? So it's this horizontal axis that all that variation really is in. The only topological difference is shown right here with this blue line, and that is Achiapola sister to Maui parrotbill, or does Maui parrotbill diverge and then Achiapola? And you'll see in the text, I think, how many trees showed this difference. But it's reasonably small, but it still gives you an idea. How confident are we in these relationships? And it tells you it's mostly the divergence times that we're not sure on with this analysis. On the left side of the figure, you see there's one set of trees, and on the right side you see there's a different set of trees. So what's the difference between this? The left is showing you the mitochondrial, a tree from mitochondrial data, and the right is showing you a tree from 12 nuclear loci, and the whole mitochondrial genes all as one. And what you'll see here is there's a lot more fuzziness in the topology, a lot more lines that show different relationships among these species. And so what this is telling us is that the nuclear tree, right, the nuclear loci, are giving us different signal, okay? So we'll talk in class about why that might be. I want you to start thinking now and give yourself a list of reasons why you might see this different pattern with nuclear loci rather than mitochondrial genes. So come to class with that list so we can work on that in team activities. There is a part on this page in which I talk about substitution saturation. Somewhere down in here we go, substitution saturation. And so I wanted to explain to you briefly what that is. So here we go. So substitution saturation is where there are multiple substitutions that have occurred at a DNA site, at a nucleotide site. And when that happens, that obscures your true evolutionary history in the sense of, in one species, you get a C to a T and back to a C. And so it looks like there's no change. However, there have actually been 2 separate mutations that have occurred there. In extreme cases, when sequences have experienced full substitution saturation, the similarity between the sequences is going to depend entirely on the similarity of nucleotide frequencies, which doesn't really reflect phylogenetic relationships, but more an organismal bias toward incorporating certain nucleotides over others. So how can you tell if you have substitution saturation? That is, that your signal is obscured by multiple substitutions at the same sites there. So think about it in terms of a hypothesis, right? In absence of saturation, you think that species that have more nucleotide differences are more distantly related, so they'll have accumulated transitions and transversion substitutions at a clock-like rate. So you can test this. You can plot the number of transitions and transversions per site versus the genetic distance between each pair of taxa in your analysis. So you would assume that applying a model of sequence evolution, right, that takes into account things like variation in nucleotide substitutions, that as you increase here in the evolutionary distance, you're going to increase in the number of substitutions and per site. So the number of transitions, the number of transversions should keep increasing, and that should increase pretty much linearly, like this green line, because we know about clock-like evolution. However, what you see is there's a difference here where transitions or transversions, which are shown in green, are showing that pattern that you'd expect to see, but transitions are not. In fact, transitions pretty much right here, let's see if I can get to the point where it starts to really level off. So somewhere around 0.48, 0.5, you see a plateau in the accumulation of transitions in these increasingly divergent pairwise species. So what's happening there to get this slowdown in the accumulation of transitions is that's saturation. That's evidence of saturation. So I think you guys can understand that now. What happened when I looked at my data, I will tell you in the text here how I looked at substitution saturation and what information that gave me, and then how that affected my analyses. So make a note to yourself, and we'll talk about what to do when you find saturation and where you would expect to see saturation, what types of sites. Make a little list for yourself, what types of sites you would expect to see substitution saturation in versus others, thinking about codon positions, thinking about, so mainly think about codon positions for now. Okay, we're going to go a little bit further. I want you to, as usual, pay attention to the figures. So our next figure, this figure 2 is actually a quite complicated figure, and I've made many variations of this for various purposes. This is the full figure with all of the information on it. So you need to take a little bit of time to read the legend. I'll give you a brief introduction to it now. Get it on the screen. Okay, so here, this data set is just showing our Hawaiian honeycreepers. It's just using data from whole mitochondrial genomes, and this is a tree that has timing for divergences on it. And how did we get the timing for the divergences? Because the Hawaiian Islands are formed over a hot spot where the plate is moving to the northwest over the top of the hot spot. We know the age of formation of Kauai. We know that Oahu was formed later, and we know when that happened from dating those rocks. Same thing, Maui Nui is younger, and Hawaii is the youngest of those islands. So what I could do is I could say that a species found only on Oahu is a species that I can say didn't exist until Oahu formed. So when I have done my analysis, I can say that, for instance, this node here, there's an asterisk here, this node could not have happened until the divergence between, it says Kauai and Maui creeper here. I'm substituting Oahu creeper because they are almost identical in terms of sequence. So we assumed that the Maui creeper was the Oahu creeper and then applied the date for Oahu to this node. We did some tests to make sure that that was a valid assumption. We also here said that here's another divergence, right? And this divergence couldn't happen until the formation of Maui, because you can't have a node going to Maui until that happens. And here you can't have Hawaii Amakihi forming until the Hawaiian island, the big island, is formed. So you can see the 3 different calibration points. We'll talk about the distance between those and how that informs your analysis. But the other thing that I want you to look at is I also have letters on here, A and B and C. Each of those identifies a major morphological type within the Hawaiian honeycreepers. So spend a little bit of time answering these types of questions. How many lineages, how many morphological types had evolved when Kauai was the only big island? Right, so before Oahu is formed, how many of those morphological types? Then think about Maui Nui. How many of the Hawaiian honeycreeper morphological types had evolved before this island existed? And then finally, which of the major types had not evolved before Maui Nui was formed, right? Which ones evolved afterward? And you can see on the right-hand side, I meant to tell you here that we've got each of the islands, and so you can see which species are found on which of the islands. You can see this group has a down island colonization pattern: Hawaii, Oahu, Maui, and Hawaii. Okay, last figure I'm going to tell you about is figure 3, and this is a map here of the substitution rates and for each of the different types of loci that I looked at. This analysis had a lot of different loci. It's one of the early studies to look at so many loci. And to have such a good dates to apply so we could figure out really exact rates of evolution for these different genomic regions. So there are 3 different data sets, A, B, and C, and then each of these colored peaks corresponds to a different sequence. So here we have nuclear regions, here we have different genes in the mitochondria, and here we have different types of sites in the mitochondria. So answer some questions for yourself. What are the fastest evolving sites? What are the slowest evolving sites? And then I want you to write out why you think those are faster or slower. And then the final question I want you to be thinking about when you're reading this is which of these figures shows a species tree analysis? And then thinking through that, what does that tell you about the different trees in the paper? And the different types of signals that you're seeing. All right, we'll talk about this on Friday afternoon.